Hey, what is going on? It is Monday, January 17th, 2022. Okay? And we're coming along, we're chugging along on our learnings of the immune system. And I have to say that I've been doing this pretty strong for the last few days. Missed only 1 or 2 days due to flukes, and I intend to try to never miss a day from now on in my learnings and in my dissemination of what I find and what's hopefully eventually uh, a cogent dissertation of my research. Um, Most of my research being internet based for now, but it's necessary, at least at this stage. So, what do I want to talk about today? Well, I've talked about the immune system. We did a small overview of the innate immune system and adaptive immune system, and the bridges between both, right? So, what I want to talk about today I want to start to get into some of the more intermediate topics of the immune system, right? So, what is one thing we've not really gone over viral activity yet, general viral activity? I will do that because that's important because viruses are one of the main pathogens categories that have to be preventively and actively prevented from progressing infection through. But I'm going to talk a little bit about AIDS today and HIV. And in the 90s, AIDS and HIV and in the 80s was like the huge bogeyman. I mean, if you compare the coronavirus of today, you compare it to AIDS of back in the day, and there was the AIDS pandemic, which received crazy amounts of news, no lockdowns, but there were some, at that time, very large and wide-scale systemic changes and effects on society, which could be considered comparable in some ways to the coronavirus situation. But anyway, let's get into the details. So what is AIDS? AIDS stands for acquired immunodeficiency syndrome. HIV stands for human immunodeficiency virus. Okay, so and how does it work? So AIDS works differently from other viruses, and on the high level, many people know how AIDS works, right? HIV leads to AIDS, etc., and you can't get AIDS from different mechanisms, but you can get AIDS from bodily fluids, etc. But let's just talk about what HIV is. So HIV is basically a virus, and it looks like many other viruses, right? Like if you look at the structure, it's basically a circular viral protein coat with spike-looking proteins around the envelope of the virus, and these proteins are involved in causing the actual infection. Okay, so how does HIV infect somebody? So HIV will infect somebody through integration of the virus particle into helper T cells or CD4 positive cells. So unlike other viruses or many other viruses, HIV is like immunodeficiency virus, meaning it causes a decline in the immune system. So it by attacking immune cells, right? In this case, T lymphocytes. So how does it, or specifically CD4 T lymphocytes? So how does it do this? Well, we'll get into that in a second. I'll jump into that in a second. But let's just talk about like how HIV progresses, right? So you have your initial HIV phase, which is your acute phase. And your acute phase is when you start to get sick, and that's within the first 2 weeks of your HIV infection. You start to get sick, and you start to see a spike in flu-like symptoms, and afterwards, and at this point, this is when there's you were first infected with a large amount of viral particulate content floating throughout your body, and your immune system is not kicked into gear yet. Your innate immune system is what's immediately working right now, but your adaptive immune system is not yet, is in its latent phase, right? This is like the course of a traditional infection. So what happens is the dendrites, dendritic cells, mast cells, however you got in this case, it would be dendritic cells, right? Because normally you're not infected through AIDS through a puncture where blood goes into your wound and then gives you AIDS. What you're infected through is STDs and some other mechanisms of fluid transfer, like intravenal through injection of needles, infected needles, etc. So you have these you have these particles, HIV particles within your body and in your bloodstream, and your dendritic cells will do what they normally do. They will grab an HIV molecule, they'll ingest the HIV molecule, they'll phagocytize those HIV molecules, and they'll bring them to your lymph nodes, bring the antigen to the lymph nodes, right? So what happens here? So basically, these HIV particles, they start to build up in your system, and you have what's called your asymptotic phase, right? So you don't, in the middle stage of your infection, HIV slowly progresses by doing what it does and infecting your T cells. And as it infects your T cells, you don't notice any disease specifically from HIV because HIV is not causing disruption to tissues that are related to your or it's not causing your inflammation and immune responses that are causing mucus and things like that, or causing additional flu and fever. It's affecting your immune cells directly, your T helper cells directly, your Th1. So then eventually what happens is, so as the AIDS infection progresses, the number of your Th1 cells decreases, and then it eventually, then later on, for years perhaps this occurs, and then afterwards, then that's when a person has AIDS, because then at that point the number of their CD4 positive cells decreases, and the amount of HIV virus skyrockets and just continues an exponential, geometric to exponential kill off of your immune cells. And again, that is not what is giving you any infection, but that's when you start to notice the prevalence of dire symptoms from infections that are like the common cold or even cancer, because your T cells are no longer, remember, your helper cells are really important in activating other cells. Your T lymphocytes, your T helper cells activate and allow T lymphocytes to proliferate and create killer T cells, CD8 cells, CD8 positive cells. Those CD8 positive cells are involved in getting rid of cancer. It also causes proliferation of the B cells and the differentiation of the B cells, which aid in creating antibodies against other infections. And now antibodies against other infections are decreased. Also, cytokinetic activity and signaling from is no longer available because your CD4 cells are gone, and the cytokinetic signaling allows your immune system to respond to an immunocompromising events, and that and it also allows differentiation, and also allows creation of new CD8 positive cells, and again, that's gone. So your immune system basically at that point grinds to a halt, with the exception of like your innate immunity or whatnot, but your adaptive immune system, which is very important to your survival, essentially grinds to a halt, and you die eventually. And again, cancer, etc., kills you too. So so let's go a little into like what is the detail, like how does this occur? And there's a lot of interesting things to note here, because the way that AIDS, the way that you get AIDS is through the HIV virus is actually shockingly complex in what it how it carries, and it feels like some people thought the HIV virus was an engineered virus, and I mean like you can look at like what it does, and you can think that, I don't think it's unreasonable initially to think that that's the case, because the HIV virus, what it does, it essentially uses its GP41 transmembrane glycoprotein and GP120 docking glycoprotein to get into your T cell. And essentially what happens is this glycoprotein touches your T cell membrane. It specifically looks for CD4 on your on cells, right? Like once it bumps into a cell that has CD4, then it will latch onto that CD4 and CXR5 cells complexes on your membrane, and it will essentially make that cell stab itself with the HIV molecule or HIV complex, right? And the GP120 will cause latching, then the GP41 will cause the stabbing, and then once the stabbing occurs, then the cell membrane is opened up, right? And you've seen, like, essentially you've seen, this is the sequence of what happens, right? Binding to the co-receptors, right? The CD4 co-receptor and its co-receptor, a CXR5. Those are what occurs, and essentially this molecule uses the yellow part, the CD4 helps bring it in, and then the GP120 stabs and creates this hole in the phospholipid bilayer or cell membrane. And then at that point, the viral capsid is released into the cytoplasmic space. Then the viral capsid itself is broken down, degraded by enzymatic activity within your cell. And then afterwards, so much stuff happens, right? Like then there's nucleic acid, there's nucleotides within your cytoplasm, which the AIDS virus uniquely uses those nucleotides floating within your cytoplasm to reverse transcribe, create DNA from viral RNA, right? So essentially it does a reverse central dogma, right? Like this viral capsid goes into your cell. The viral capsid has proteins, has its own the actual viral RNA, and then these assistant proteins called integrases and reverse transcriptases. The reverse transcriptase creates a copy of your DNA, creates a copy of the viral DNA from the RNA, the complementary RNA, and then it creates double-stranded DNA. Then the integrase goes within a cell, right? The integrase goes within the nucleus of a cell, and once that happens, it's then the integrase, along with the new double-stranded viral DNA, cleaves a part of your own DNA and inserts that viral DNA double strand into the DNA, your DNA, and just it creates this connective region. So what happens at that point, right? So at that point your cell will try to synthesize DNA. We'll try to synthesize proteins through transcription and translation, transcription, etc. The standard central dogma. It will run across the DNA of the virus and then synthesize viral components. The viral components then are brought to, they're synthesized within the endoplasmic reticulum through the standard ribosomal activity. They're brought to the surface of the cell, and then at the surface of the cell they're cut up into pieces and by an enzyme the virus contains, and at that point so integrase, just to let you go back to what integrase does, integrase is doing the actual cleaving of the DNA, right? Integrase in HIV opens the DNA up and then does that, and then that's where that process starts. But what happens is once the proteins, this is the whole system, the whole life cycle of HIV. Here, the integration occurs, replication occurs, and then assembly occurs at the cell membrane when the protein, the viral protein, which contains like a long string of all the proteins that the virus needs combined together, exits the ER, is brought to the cell surface, and then creates a new HIV particle on the cell surface and buds away. Okay, so basically this is kind of like the actual scanning electron microscope image of this, but budding of the particles, the viral particles off the cell wall of the T helper cell is what's happening. Okay, and eventually, this process, either the rest of the immune system that's active at that point takes note of what's happening, or this budding kills off the T helper cell. Either way, there is the T helper cell will most likely die after it's created many more HIV particles, right? And it's interesting looking at the HIV particle. Like, obviously you can't see individual atoms, but you can kind of see like the structure of that particle. So once that particle then gets created, it does its thing to another T helper cell. The T helper cell, in fact, now the T helper cell has internal defense mechanisms against the action of these HIV molecules, but the action will, this pyroptotic action, as it's called, inflammation action, and apoptotic actions, your standard like cellular watchdogs, will kill off the T cell, right? But the interesting thing is, and not so far in the distant past, it's been discovered that even though the T helper cells die that are infected, most likely die, the actual reduction in immune cells that occurs is not due to the infective cell's death, but it's due to uninfected cell death. And uninfected cell death occurs, and it's pretty sad, but uninfected cell death is what essentially is, it seems like, an evolutionary mechanism to prevent the exponential spreading of AIDS. And how that works is that your cells, your uninfected cells, actually succumb to signaling that occurs because of pyroptotic inflammatory activity within your existing cells, within your existing infected cells. So your T helper cells essentially have an inflammation cascade which occurs within them, which occurs because of an enzyme called caspase, which, when it detects viral transcripts, it starts causing inflammation, and that affects the cell and causes cell death, pyroptotic cell death. But it also creates a large concentration of interleukin B. And interleukin B is a cytokinetic actor. This is a cytokine which induces inflammation in other cells. And what happens is, once the T cell dies and ruptures, the interleukin B, it'll be emitted anyway, but it will emit from this T cell infected, and it will then be it will interact with other helper T cells, and then at that point, those cells have their own inflammatory responses. So it's kind of like a chain reaction that occurs, like a cellular chain reaction. And that is what the primary actor in the contraction of AIDS and conversion of AIDS to HIV, or contraction of AIDS from HIV, is indicated upon. The reactive pyroptosis of T helper cells that are uninfected from infected T helper cells. So, very interesting to me. I didn't actually know. This was actually fairly recently discovered, and there are many ways to prevent AIDS infections from increasing, but obviously you can't do in a 20-minute video what it's taken researchers a long time to do. So this has been a long video, but I thought it was really interesting to get the higher level mechanisms of AIDS and HIV kind of lined out. We'll be changing gears next time, but at some point it might be worthwhile to go into all the details. It's a very complex cellular cascade on the cellular landscape and biochemical landscape that's involved in this process. So yeah, we'll talk about that at some other point. All right, I'll see you later. Have a good day.